Hi, everybody. Welcome. Thank you so much. I think you can hear me all right. Yes, give me a thumbs up, anybody. Lovely. Yes, so I'm so happy to be welcoming you here to this first webinar of UAC in this year of 2022. Already can't believe it's already 2022. Some of us are back at working from home due to the pandemic. So if you are from home, I guess there's a benefit of having these things online still. And I hope that all of you had a very relaxing and nice holiday time over the change of the year. Today we have another two of the PIs here at our center, and I'm very, very happy to introduce actually the first PI that comes from the other university that we have here in Uppsala, which is the University of Agricultural Sciences. We are going to start with Susanna's talk, and then we'll have a little break, and we'll continue with Johan's talk afterwards. As you know, there is time for questions and answers, I hope, in both of the talks. So if you have any questions, you can either write them on the chat, or you can then raise your hand and open up your mic when the Q&A section starts. Susanna is going to talk to us about something that we've been trying to bring into the center for quite some time, and we think is important, which is the concept of One Health, and like the development of AMR not only within human medicine, but also the veterinary medicine and in the farms. So she's going to talk to us about how they are working with to understand the dynamics of AMR, in particular Swedish pig farms. Susanna, the floor is yours. So please, can you share your screen and then you can start. Thank you everybody for being with us. We are about 20 people now, which is great. And remember, you can write your questions in the chat, or you can wait to the Q&A section and raise your hand. Thank you, Susanna.
Thank you very much. Yes, I'm from the other university, the Swedish University of Agricultural Sciences, or SLU as we call it for short. This is a new project, so we haven't done much yet, and I'm happy that I'm the first speaker in this session, because otherwise it would look really strange with a lot of results and stuff, and then I'm just saying what we're going to do. But this is a new project with this round of PhD students in the sort of second round in Uppsala Antibiotic Center. So this is the title of my talk. Our project is on exploring the dynamics of AMR and we'll use pig farms, but I'm going to talk a bit first about the One Health aspects and the global use of antimicrobials in livestock, and then the use of antimicrobials and the antimicrobial resistance in Sweden. So this is a bit of the One Health setting, and then I'm going to talk a bit more about what we're going to do in our project. The One Health aspects of antimicrobial resistance are quite obvious. We treat people and animals when they are sick with antimicrobials to cure bacterial diseases. This is done differently in different countries as regards what we call prudent use, how much you use diagnostics before you treat, and how much you think about broad spectrum antibiotics versus narrow spectrum antibiotics, and if people who prescribe, if there is a prescription requirement, which there is in Europe, but in a larger context it might not be, how much the prescriber thinks about AMR and the risk of AMR and so on. And this is a graph from the yearly reports about antimicrobial use in animals in the EU or in Europe. And these are for the different countries. You can see that it's not just the European Union, it's the other European countries as well. These figures are corrected. It's a PCU, that's population corrected units, because animals are very different sizes. I mean, people are also of different sizes, but not as different as one chicken compared to one cow. So one treatment for a cow will be so many more kilograms than a treatment for a chicken. So this is why these figures are corrected, so that it would be more or less comparable. It's always difficult to compare use of antibiotics because the different substances vary with how much the substance weighs and so on. But this is an effort that's done yearly to show how much is used in the different European countries. And then there's also some figures on antimicrobial resistance, but this is the usage. And as you can see, Sweden has a very low usage. Of the EU countries with the lowest, but we have Norway and Iceland who have a lower usage. And as I said, this is corrected for animal population. So this is not just because we have small animal populations in these countries, it is because we use very little antimicrobials for our animals. And the policy is to use it for treatment, but not for anything else when needed. Sick animals that need antibiotics should have them, but not healthy animals. And this is something that's now becoming more and more obvious or taken for granted in most countries, but it used to be that we were the strange ones who didn't want to feed our animals with antibiotics. It's changed during the past decades. In Sweden, this is the sort of motto of Swedish veterinarians: healthy animals don't need antibiotics. This is just to provide you with the background why we need to do this type of research in Sweden, because we actually don't have so much antimicrobial resistance. The background noise is not so much, and we really don't use antibiotics routinely if we can do something else. So there's a big focus on preventive animal health and to avoid getting into a situation where you need to treat the animals. These are from the Swedish reports, the Swedish yearly reports on antimicrobial use and AMR in people and animals. So these are One Health reports. They have been so for a long time. This is a Swedish farm report that's produced yearly by the authorities within public health and veterinary medicine. And it compares the sales of, in the left graph, it's fluoroquinolones, macrolides, lincosamides, trimethoprim, and sulfonamides and tetracyclines for human and veterinary medicine. It's kilogram active substance, so it does not. This one does not take into account the number of individuals being treated and how large they are. We have more people than animals in Sweden, than livestock anyway, but the livestock are heavier. But as you can see, there is more, the total amount of antibiotics used in humans is much larger than the total amount used in animals. And there are various reasons for this, but I just want to show you this, because there's a lot of talk about the global use in animals. There's so many antibiotics being consumed by animals, and there's so much AMR in animals and so on. This is not the case in Sweden. And this is why our research has a sort of different background, and we come from a different angle, which is very useful from a research point of view as well, not just from a One Health point of view. And this is also from the Swedish farm report. This one is corrected per estimated kilogram biomass. It compares the use of some antibiotics in animals and in humans. And again, the red bars are animals and the blue ones are humans. And you can see that there is a lot more in humans. And Sweden does not use a lot in humans, in human medicine. Sweden is regarded as one of the countries where there's a prudent use, and we're sort of known for a good preventive health work and a restrictive use of antibiotics in human medicine, but the veterinary medicine stands out. And then you go from the reason that we're monitoring the use is, of course, that this is reflected in the resistance figures. Nationally and globally, there's a lot of correlation between antimicrobial resistance and antimicrobial use in different species and different substances and so on. And in this report, the Swedish report again, there are comparisons between E. coli from different clinical specimens from animals and people. So these are clinical specimens. These are expected to be the resistance prevalence in these specimens, or in these bacterial strains are expected to be higher. The levels are expected to be higher because these are from sick people and animals who may be in an environment where there is a lot of antimicrobial use. They might already have been treated and so on. So this is not the background anymore. This is the clinical situation. But as you can see, We use quite, the preferred drugs are beta-lactams, so ampicillin is used quite a bit in animals, and you can see there is some resistance there. Ciprofloxacin, we use enrofloxacin in animals, but that's a last resort, so we are only allowed to use it for animals if we can justify it by a culture and susceptibility test that shows that this is the only one that we can use. Then we are allowed to use quinolones, so the ciprofloxacin resistance is expected to be fairly low among animal isolates. The same goes for cephalosporins. We're also only allowed to use second and third generation cephalosporins if we can justify that by a culture and susceptibility test. So there's generally quite a low level of resistance. And then there are some other substances that you can see there is the stuff that we use is reflected in the resistance patterns. It's the same for people and animals. So why is this interesting? Yes, it's because we want to use Swedish pig farms as a study setting. We think that this will be sorry, I got distracted by the chat. I thought there might be something there for me there. We think that Swedish pig farms will provide a very good study setting for looking at AMR dynamics because we have a low overall AMU, that's antimicrobial use, and a low overall antimicrobial resistance levels. That Swedish pig farms have good biosecurity, so there's the risk of bringing in and out resistant strains would be lower than in other situations. It's not non-existent, but it's a fairly low risk. It's a structured production system, so you can follow animals throughout their lifetime in these herds, and you can follow a group of animals from when they are born until they are go to slaughter. And there is sufficient variation between farms. The Swedish pig farms are fairly similar as regards this production systems and the structure and so on, and the regulations, of course, the national regulations about animal welfare and what you're supposed to do and how you're supposed to record your treatments and record everything that goes on with the animals. But there's also some variations as regards size and management and so on. So there will be a low background noise of resistance and not very much use of antibiotics, but also a fairly low background noise of some diseases that are present in other countries, but we have actually eradicated in Swedish pig farms. There are closed environments because the biosecurity is good and they don't bring in new pigs all the time. There is sufficient variation between the farms. So we think that these will serve as a good type of living labs for this. And then we want to look at the AMR dynamics. We want to look at the antimicrobial use in these farms. We also want to look at the animal health management factors, and these are various aspects of animal health management. That could be vaccination programs, it could be cleaning and disinfection routines, how they separate different categories of animals, if they have sectioning, if they have extra hygiene routines between different stables where people change their clothes and so on, how they handle their external biosecurity, how they avoid to bring in diseases via feed, deliveries, people visiting veterinarians and so on, the general management, how they prevent disease and also how they treat disease. But there's also the general animal management, how they make sure that the pigs are comfortable and that they get good feed and good quality water and that they have as good a life as possible. So this is the animal welfare aspect that also affects animal health. Animal health can be seen as part of animal welfare. But also the animals themselves, their genetic background, and this is usually very similar within the same pig farm and also within the entire pig population in Sweden, but there are some variations. But also general, they are affected by how they are brought up. the farmer attitudes, and the farmers and their staff, how they see preventive animal health and how they see the animal, how they handle the animals. That affects the general situation of the animals and also their risk of disease, and the risk of disease again affects the use of antimicrobials and the presence of antimicrobial resistance. But it's also the physical environment that can affect how much resistant bacteria persist in the environment. This is about hygiene, but it's also about other factors in the physical environment. So we're going to look at all these factors in various ways. So we're going to sample these farms, sample the immediate environment around the animals, because we don't want to sample the individual animals, because then we will end up with all these individual variations and so on. We want to know the farm environment, so that's indoor environment. We're going to apply metagenomics on this, so try and identify or characterize the resistome of these samples, and then we apply bioinformatics on the metagenomic results to characterize what is in there, what resistance genes are in there, and what's the composition of the general microbiome and the resistome. There's going to have to be a lot of validation of this workflow. So our PhD student has just started this work. The sampling technique, where to sample, how to take the samples, how to transport them, what type of metagenomic methods would be most suitable, which would be use one or several in combination, and how to apply the bioinformatics in these different situations. So this entire workflow needs to be validated and assessed, and we need to produce a good protocol for this. It's also the data collection, and I mentioned all these different risk factors, these aspects of the resistance dynamics in the farm that we need to collect data on, and they will be in different formats. And we need to design questionnaires for the farmers. We need to interview the farmers in a structured way. We need to take samples in the physical environment. We need to look at the farm records, and we need all these agreements with the farmers so that they allow us to do this, both the sampling and the data collection. So this is a lots of work before we actually start collecting the data. And we have just started the planning because we have just started the project. And then we will do epidemiological analyses on these data on the farm resistome and the risk factors, and try to get closer to the actual dynamics in addition to antimicrobial use. What else is affecting the increase or decrease or persistence of different resistance genes in the farm environment? So challenges here include the sampling and sample transport, because we have farms that are in various places in Sweden, various locations in Sweden, and they are built up differently, and we have to sample repeatedly. We need to decide whether to go there ourselves or whether we're going to engage veterinarians or the farmers themselves to take samples, and the samples have to be transported. And this might include needing to have them on dry ice, and how would this practically be achieved if the farms are far away. We also need to look at whether we should use long or short reads or a combination in the metagenomics analysis. We need to, sorry, that's a spelling error. We need to look at the representativity of the samples in the farm, and then how many farms do we need to make this statistically relevant. Because there's a trade-off here with the number of farms, the number of samples, and the in-depth study that we can do. There's also the issue of data accessibility and validity and detail. How much can we get from the farmer? How much can we get from registers if the farmers allow us to get them from registers, and so on. And this data will be very heterogeneous. So how can we put them into a format that way we can actually perform standard analyses, or do we need to adapt that somehow? And in the future, of course, we want to bring this further. This is just a PhD student project that we start with. So we want to bring these data into further advanced modeling methods with other collaborators that we need to engage in our project. So we're currently applying for larger funding to bring this project into another phase already. We would also like to look at new resistance genes. This is sounds easy, but it's not always that easy, and it needs to be validated. We also would like to involve social science, because the behaviors and attitudes and drivers of the farmers themselves and the veterinarians are known to affect this in very many ways, and some of the data that you cannot collect numerically can be captured in how the farmer sees their farming and their animals, and also how the veterinarians work and see their work in the context of antimicrobial resistance. And in the future, we would also like to apply this approach to other systems, other environments, with other animal species and with humans. So we have high-flying plans, but we're starting with this small project now. And our project team involves me as a PI, and also my colleague Elizabeth Rayala, who is a veterinarian and a PhD. She has a PhD in veterinary microbiology. We have our doctoral students, our PhD student Valeria Ladehina, who has just joined us in SLU. We also have Associate Professor Johan Dixved, who is a microbiologist, and Professor Erik Bonkam-Rudloff, who is a bioinformatician. And by that, I think I stop and invite you to ask some questions.
Thank you so much, Susanna. That was great. Great description of what you guys are going to do. We open now Q&A session, short Q&A for Susanna. So we have a question from one of our PhD students. Philip is saying, thank you for your talk. Is there such a strict control of antibiotic usage in Sweden on farms? And the level of AMR is kept relatively low and seems constant from current knowledge. Is there any risk of AMR development at all, as long as the regulations and the control are kept very strict? Or is there some catch-22 behind keeping AMR levels relatively low?
I think the operative word there is relatively low, because this is in relation to whether other places where the levels are much higher. The thing is that all antimicrobial use selects for AMR. So even if it's low use, and if we don't use antibiotics at all, then we don't need to worry about AMR, because the only worry about AMR is that it renders antibiotics useless in some instances. So there is a catch-22 somehow, because we want to be able to use antibiotics to treat sick animals and sick people, but every time we use them, we trigger AMR, and it will eventually it will keep increasing unless we can find some magic way of avoiding this. But it can also wane in the long term. But if you keep using it, if you keep using the same substance, AMR will just increase, even if you don't use much. So yes, we do have AMR, and it does increase, but it doesn't go as fast as in some other countries where the regulations are not so strict.
Great. He's also asking, are the tests for antibiotic susceptibility in animals same as those in humans?
Yes, we use the same tests. I mean, these are bacteria, so the lab work is the same. Sometimes the cutoffs, the clinical cutoffs can be different. The interpretation of the cutoffs can be different. I'm not a expert in pharmacokinetics and dynamics, so I don't really want to go into how they differ, but they can differ in some instances.
Mm-hmm. Interesting. Great. If anybody has any other questions, I was, Mingwan, I can ask you something, Susanna. So it feels like this is a complete new way to look at AMR here in Sweden, if I'm not mistaken. Like before, the way that they have been monitoring it somehow is by taking samples of already sick animals and then seeing the percentage of resistant strains. Is that correct?
No, they're actually doing both. So we have a long-standing, and the Nordic countries, I should say, have a long-standing sort of monitoring of AMR. So you take both indicator species, E. coli, enterococci, and they are collected regularly on farms from the farm environment or from healthy animals, just to monitor the general background. So that's the sort of monitoring, and then you have the clinical isolates, which will be which are expected to have a higher level of resistance.
All right, and so I assume also that the level of antibiotics being used in Sweden from the early 80s, 86 I think is when the ban came in, and then up to here. Is there old data that says there was much more prevalence on AMR genes in the farms before versus now?
I don't think there was much more prevalence. I'm trying to remember. I can't say that some resistance levels have gone down because we don't use the antimicrobials anymore, and some have gone up when we've brought in new antibiotics. You can see that once you bring in a new substance and you start using it, resistance levels will go up. In some countries they monitor salmonella isolates, but since we have a different attitude to salmonella in Sweden, so in Swedish animals we try to eradicate the bacterium, or not eradicate, but we control salmonella. I shouldn't say we don't think we can eradicate it totally, but we have a very strict salmonella control, so we try to eliminate it from herds where we find salmonella. And then the resistance in salmonella is not a clinical problem in animals because we don't treat them. We try to get rid of the bacteria instead via other means. So I'm not sure whether we actually did have more AMR, but we had a lot more general use. And the reason for banning feed additives was not AMR. It was just that the consumers thought that it was sort of disgusting to feed animals with antibiotics, or the farmers realized that the consumers might start worrying about us feeding animals with antibiotics.
I understand. So it was more not just the direct result on the resistance prevalence, but mostly that it's an extra chemical that goes into the animals.
Yeah.
And potentially...
So it was a general thought that farming should be clean and not use a lot of chemicals and so on. And so the farmers initiated this, and then it was a sort of general ban on antimicrobial feed additives for productive reasons. But then when we removed these low-level antibiotics in the feed, the animals got sick. So we realized that we had actually been preventing diseases. I wasn't part of that, but we had been preventing diseases. So then there had to be this big focus on preventive animal health just to keep the animals healthy.
So they changed the procedures and the way they work around the farms to keep healthy animals rather than having to preventively treat them. That's very interesting. Very nice.
So now we're sort of boasting about this, how good our preventive animal health is, but that was actually a necessity. When we banned the feed additives, we had to do this, and in the end it was a good thing, but back then people probably were a bit worried.
Yeah, obviously. That's right. Anybody else has any questions? You guys can raise your hands if you want to. Oh, Professor Tanvir, he has
Yes.
Yeah. Hey.
Hi, thank you.
Welcome.
So, I am Professor Dr. Tanvir Rahman from Bangladesh Agriculture University. I am working on AMR along with a few other things. It's a pleasure to be here. Suddenly I came across the link and registered myself and joined. So I think it's okay. I just wondering, I mean, it's a nice proposal, but since it's focused on One Health approach, as I understood. So personally, I believe it will be good if you are able to include some of the samples from the pig, and also, if possible, the people working in the farm, so that you have a complete coverage of all the components of One Health. Otherwise, it's really a nice study. Thank you very much.
Thank you. Yes, that's something that's been done in some projects. We've had PhD students in SLU who've done that in not in Sweden, but in some other countries where people and animals their lives are more intertwined, I should say, where the animals are closer to the households. So we've had some PhD students who have looked into sampling the farmers and their animals. And there is obvious links because they exchange bacteria, and then if they are resistant, there will be an exchange. I think it would be very interesting to do more of those studies, but I think it's also a challenge as regards ethical permits and how people feel about being subjected to along with their animals subjected to such a study, because there is a lot of interesting aspects where transmission of bacteria, resistant bacteria, don't just go from the animals to people, but the other way around. So it would be interesting to know how much antibiotics you use in people, and if they transmit this to the animals and so on. It's known that in countries where you have a lot of pets, that you can get, if you use antibiotics in people, they can actually transmit resistant bacteria to their dogs and so on. If you have the antibiotics in your household, the animals, and the animals are also in the household, they will be exposed to the antibiotic, and this will also select for resistant strains in their flora and in the human microbiota. So there's a lot of exchange going on there, and I think there's lots of projects that could be interesting to do. We won't be able to do it in this project, but it's important stuff that we need to look at, especially when people and animals live close together.
Mm-hmm.
Okay, yeah, thank you very much. Thank you.
Great. Thank you so much, Susanna. Is there any other questions in the chat? Nothing here, no one opening their mic either. I think that is that for now. Of course, Susanna, you are one email away, I guess, if anybody has. I really like also that you mentioned that you want to potentially, look for new genes, which also needs new techniques, and also you want to expand perhaps to a more social aspects of the project, and that calls for collaborations and people that are experts in doing those stuff. Maybe they can reach out to you as well and maybe talked about it. So that's great that you have this bigger outlook on the project, not just the samples and the results of it. Thank you so much, Susanna. Thank you all of you that paid attention to Susanna's talk. We are going to now have a break.